Hi everyone, my name is Ankatrin and I would like to welcome you to the video abstract of molecular and physiological adaptations to low temperature in Thioalkalivibrio strains isolated from soda lakes with different temperature regimes. So soda lakes are dual extreme environments with a high pH and a high salinity, but next to those also temperature can be a supplementary stress factor to which the microbial communities need to adapt. So, for instance, soda lakes in Siberia are exposed to strong seasonal temperature fluctuations, with warm summers and freezing winters, whereas soda lakes from the East African Rift Valley encounter a warm and stable temperature throughout the year. One of the most abundant taxa found in soda lakes are the sulfur-oxidizing bacteria of the genus Thioalkalivibrio. Its strains have been isolated from soda lakes worldwide, also from lakes with seasonal freezing temperatures. So therefore the question arises of how they adapt to low temperature. To obtain an answer to this question, we took the strains Thioalkalivibrio versutus, AL2, isolated from a soda lake in Siberia, and Thioalkalivibrio nitratus, ALG2, from the East African Rift Valley in Kenya. We cultivated both strains under controlled conditions in chemostats at 10 and 30 degrees, and thereafter we compared their membrane lipid composition, their glycine betaine content, and also their gene expression by RNA-Seq for both temperatures. We found that the Siberian strain AL2 grew much better at 10 degrees than the Kenyan strain ALG2, as AL2 had a shorter lag phase and a higher maximum growth rate at this temperature. Looking at the membrane lipid composition, we saw that both strains increased their unsaturated fatty acid content, which is a well-known mechanism to maintain the membrane fluidity at low temperature. And gene expression data revealed that the FAB pathway appears to be a major desaturation mechanism under this condition. Comparing the concentration of the osmolyte and cryoprotectant glycine betaine at both temperatures, we saw that both strains increased it at 10 degrees. But AL2 actually reached a 3 times higher concentration than ALG2 at low temperature, providing the Siberian strain with a major growth advantage. Low temperature can cause an increase in the negative supercoiling of DNA, an increase in the stability of secondary structures of mRNA, and can lead to protein misfolding. To counteract these effects, genes encoding helicases, chaperones, and proteins of the transcriptional and translational machinery were generally upregulated in both strains growing at 10 degrees. Another negative effect of low temperature is the increase in oxidative stress. Interestingly, both strains highly upregulated their vitamin B12 biosynthesis pathway, which has also already previously been shown upregulated in Thioalkalivibrio under arsenite stress. For the sulfur metabolism, we could see that both strains decreased their gene expression of the second half of the sulfur oxidation pathway, leading to an accumulation of sulfur granules in the chemostat reactors at 10 degrees. And looking at the genes of the energy metabolism, we could see an increase in the expression, reflecting an increased demand for ATP to perform adaptations towards low temperature. Finally, genes for the intracellular signal transduction were differentially expressed, and interestingly, AL2 upregulated flagellin expression, whereas ALG2 downregulated it. So in conclusion, we can say that the Kenyan strain ALG2 possessed a similar adaptation response to low temperature as the Siberian strain AL2. The exception was the higher glycine betaine concentration in AL2, which identified this osmolyte as an essential actor in the cryoprotection in this strain. Therefore, we hypothesized that the adaptation to low temperature stress invokes actually multiple basic stress responses in Thioalkalivibrio that trigger inherent protection mechanisms against, for example, oxidative stress. So even though ALG2 does not face cold stress in its environment, it must cope with other common soda lake stressors, such as high UV radiation or salinity. Therefore, these adaptations are also maintained in strains living in absence of low temperature pressure. Thank you very much for listening.